Bismillahirrahmanirrahim. My name is Adnan Tahir. I am PhD student from Beijing Institute of Technology, China. Here we will discuss about the PCR primer designing. We can see that how we can design the good PCR primer. In this lecture, first the major contents of these lectures are the first is the PCR polymerase chain reactions. What are the major steps of the polymerase chain reactions and how the primers anneal to the PCRs double stranded DNA for the specific amplification of that target DNA and how we can design the good primer by using which guidelines and tricks. And then we will discuss about the primer designing tools which are available online or we can also download to design the primers. At the last in this lecture we will discuss about the brief summary of these lectures, and then we will go to the online tools for designing the primers. The first, PCR, polymerase chain reactions. As we all know that PCR is widely held as one of the most important inventions of the 20th century in molecular biology. It was invented by the Carey Mullis in 1985. By using this PCR, we can amplify the even small amounts of the genetic materials to identify manipulate DNA, and we can also detect the infectious organisms which are including the viruses that can cause the multiple diseases including AIDS, hepatitis, tuberculosis, and we can also detect the genetic variations including the mutations in human genes and numerous other tasks, and it is the very important in molecular biology. And the basic PCR working is that I hope we all know that we have hear about these steps, and but we are discussing here with respect to the primer designing, and we will discuss the some basic steps. So the main steps of the PCRs are the first is denaturation, annealing, and extensions. First, first step denaturations. Here the genetic material DNA is denatured from double stranded to single strands. And then the next step is the annealing, where the primers are then annealed to the complementary regions of the single stranded molecules. And the final step of the PCR, that is including the extensions, and then after the annealing the primers, they are extended by the action of the DNA polymerases to finish the final quantity amplified DNA. All these steps, including the denaturation, annealing, and extension, these are the temperature sensitive. And the common choice of the temperature, that is the 94 Celsius centigrade for denaturations, around 60 Celsius centigrade for the annealing temperature, and the 70 Celsius centigrade for the extension, respectively. Now we will see that the primers. What are the primers? The primers, with by definition, is a short single stranded DNA sequence that is used in the PCR technique. In the PCR method, a pair of the primers, that are the forward primer and the reverse primer, which are used to hybridize with the sample DNA and define the region of the DNA that will be amplified. Here we can see that the primer, that are the sequences, that is the forward primers and reverse primer, and that can anneal to the template DNA from 5 to 3 directions. And here we will also see that the we have to when you have the DNA sample, you have to define the which region you have to amplify, so that you can design the primer according to that region you want to amplify. That is the main important point for this PCR primer designing. Because if you are just seeing the whole DNA sequence, that may be the 100 to 10 kb base pairs, and you don't know which region you want to amplify, and you are the primers that are according to different regions, That is the total wastage of the reactions and your time and money. So that's you have to know about the which region you have to amplify through the PCR reaction, so that you will design the primer according to that region. And here we see the primer designing. And for the good primer designing, that is essential for the successful reactions, and the important design considerations are a key to specific amplifications with high yield, and the preferred values for the PCR primer designing that are indicated are built into our product. The first, the tricks, the guidelines for the good primers. You have to know about this one. The first we are the primer length. So first, if you don't know about the how we can design the primer, the first question arise from your mind that what length should be the primer. It is generally accepted that optimal length of PCR primer is from 18 to 22 base pairs. That may be the 24 base pairs. These are the optimum length of the primer. That can also maximum, we can also maximum 30 base pairs, so that these are the optimum conditions for to attain the high yield of the PCR. And this length of the primers is long enough for adequate specificity for the DNA sample, and it should be the short enough for primers to bind easily to the template at the annealing temperature, so that your primer should be long and small enough to bind the specificity and easily. If your primer is too long and it is too short, it will also lose the easily binding and the also specificity for the specific template, so that it is the. And the next, we will consider the primer melting temperature for the designing the primers. So Tm, by the definition, it is defined as the temperature at which one half of the DNA duplex will dissociate to become the single stranded, and indicates the duplex stability, and when it will from double stranded to become the single stranded DNA sample. And then the primers with melting temperature in the range of around 52 to 58 centigrade, or sometimes 50 to 60 centigrade, generally produce the best results. We can also get the more than 60 or around than 50, but we can also can't get the best results for that those primers. And sometimes the primers with melting temperature that is above 65 Celsius centigrade have a tendency for secondary annealing, because they can start the secondary annealing of this PCR product. So that's the melting temperature of the primer is the also very important for the good primers. The third we can the major is the GC content. The GC contents, that is the number of guanine and cytosine in the primer length as a percentage of the total basis of the that should be around 40 to 60% of your total length of the primers. If you have forward or reverse, forward primer is 20 base pairs, that means if you and total number of GC are 10, that shows the 50% of the GC contents. So that's these should be the around 40 to 60% of your total primer length. And the next is the GC clamp. This is also the very important for the good primer and for successful reactions of the PCR. So the presence of GRC bases, guanine or cytosine bases within the last 5 bases from the 3' end of the primer is known as the GC clamp, which helps to promote the specific binding at the 3' end due to the stronger binding of the guanine and cytosine bases. Because guanine and cytosines, they share the triple bond as compared to the adenosine and A and T, they share the secondary bonds, second bond. double bond, and we in GNC they share the triple bond. And as we can see in the figure that as the last five bases are the shown, and we can see that there is a GC clamp showing the presence of the C, that maybe also the we can also the add G and C in different combinations. But if their G or C is the more than 3, that should be avoided in the last 5 bases at the 3' end of the primer for to lose its specificity. So this is also the very important GC clamp, the presence of the guanine or cytosine bases within the last 5 bases from the 3' end of the bases. And next is the primer structure. And primer structures, and when we are designing the primer structure, the structure of primer should be the relatively simple and contain no internal secondary structures to avoid the internal folding, because if the primers contain the internal secondary structure, it can fold by itself, so that it can also affect on the yield, because it will not annealing to the PCR product and template DNA, and so that the reactions will fail and we cannot get any yield from the PCR reaction. And one also needs to avoid the primer annealing, which creates a primer dimers and disrupts the amplification process. If your primers contains the multiple complementary oligonucleotides, for example A and T, A, T, G and C repeats, so that it will create the primer dimers and disrupt the amplification process. So this is also very important for the primer designing. So these 5 points that are we have I have discussed, these are the basic points that we need to be considered for the good primer designing. And here we see that what are the, and we have to know about the which are the tools that we can use for the primer designing. A number of the primer designing tools are available that can assist in PCR primer designing for new and experienced users like. So that these tools may be reduce the cost and time involved in the experimentations by lowering the chances of failed experimentations, which are the some most of the used tools are mentioned here. The primer 3 web, that is use the online servers that can help and very easy to use for the new users. And primer 3 plus, that is also advanced form of primer 3 software. And the primer blast, this is also very easy and web based tools that can easily used for the primer designing. And the primer 3 is also available, only primer 3 is also available for to download, and we can also download the library so that it can also help to design the primers without having the internet facility. At the last of the lecture, we have summarized, these are the some guidelines and tricks that we have to remember for generating, for designing the primers. So that if we don't need the software, so you have to know about these properties, and by knowing these properties you can also design the primers without help of the any tools or softwares, and by only drawing on your notebook. So you have to, for example, first the length of the primer, that should be the 18 to 24 bases. If you have the total sequence of the DNA samples, you can count the number of the bases from the start for the forward and reverse primer, and that should be optimal from 18 to 24 bases. And we can also give the maximum 30 base pairs, so that for the GC contents we can count the number of the guanine and cytosine in the sequence, and these should be around 40 to 60%. And then we also count the start and end with 1 or 2 GS guanine or cytosine pairs, so that we can check manually by the how many guanine or cytosines are available at the end 3' end, so that will help for the specific binding. And then the melting temperature, that is around 50 to 60% of the primers. And then the primers, and we can if we are counting by self, we can count the melting temperature of the if we are counting the one guanine, that guanine or cytosine, we can count their temperature 4 Celsius centigrade. And if we are having the ART, then we can count their melting temperature at 2 centigrade. So we can count the total number of bases, and there we count the total number of the temperature of these bases. And then primers pairs should have the TM within 5 Celsius of each other. And when we have the forward and reverse primers, the primers pairs, they should have the TM almost similar TM. These are the good ones. For example, we have the forward primers with the 58 Celsius, and the reverse primer also should have the around 58, and maximum these TM should be within 5 Celsius of each other. And the primer pairs should not have the complementary regions, so that if we have the primer pairs have the complementary regions, it can lead to the primer dimers and hairpins, and they can bind to itself, and so that they will not, if they are not bind to the target DNA samples, so that it will not give any yield. Note, sometimes you are including a restriction site at the 5' end of your primer, so then you should include the around 3 to 6 base pairs clamp should be added upstream in order for the enzyme to cleave efficiently. For example, GC, these are the clamp for the restriction side your sequence. Sometimes in cloning experiments, we have to need that the enzymes restriction side, so that we have to add these clamps for the enzymes to cleave efficiently. And these are the some basic guidelines or tricks you must know about the designing the primers or learning about the how primer works. So you have to need about that. These are the optimum conditions for designing the good primers. These conditions may be vary. If you have, after designing the primer, if you are failing the experiments, you can check the primers again according to these conditions, and your which region you want to amplify, and you have to check that. If you are still not getting that your yield, you can change the annealing temperature in PCR reactions. If you are mentioned that the around, you can use the around 48 to 54, 56, 60, maximum 62 centigrade, and we can change the conditions of annealing and extension, so that we can also get the yield, good yield from the PCR. If we are still not getting the PCRs annealing with the target DNA side, then you can also again you should look about these primers, and you can design again with the careful, and we can you can use by on or any other tools. At the end of this lecture, I hope that you can design the primer now. Congratulations, you can design the primers now by remembering the by utilizing these guidelines. So now we can go for some basics know-how about these tools that are available for online, easy for new users, for that we can design, how we can design the primer by knowing according to these conditions are using the default meters. So we can go from here. Here you can see that for example, Google, you first have to get the sequence of any gene or nucleotide sequence. For example, we just enter here the NCBI. You can use any resource for nucleotide sequence, just like we can mention, for example, base 1 gene from the NCBI resource, so that we can gene the first one, beta secretase enzymes. So we have selected this one, and after getting this is the summary, this is all the information about this gene, so that these are the genomic regions, transcripts and products. So we go to the directly FASTA, go to the nucleotide FASTA sequence, and here is the nucleotide sequence of this gene. So for example, we can use this gene's sequence. This is enough for only used to check about the software working. So we have to copy this one, just like I have this one, the web page URL of the primer 3 web tools, and this is the primer 3 plus, and we will show about these two tools and how they work, and how you can change the conditions for these. For example, this one, and you can paste. This is the interface where you can put the sequence, and you have put the sequence that you want to amplify by using the PCR reaction. First, you can see that the primer 3 web, and select the task to be for primer selections. Here, this is the generic. Generic means the simple one that is mostly used for the PCR reactions. And here you can see the other options like pick sequencing primers, that the primers that can be used are sequencing, and pick primer list, and the check primers to validate the primers, so that we only select the generic primers. And then these are the sequences, and this is the, we can select the mispriming library, repeat library. We have to, when we have to check that if there are any mispriming due to their internal secondary structures are the repeats oligonucleotides, so we can select the humans, or you can also deselect or any specific. So we have selected the human, and here we can see that the pick left primer for forward primer, and the pick right primer, reverse primer, so that these are the selected. And here you can see that the sequence ID, that you can put the any, for example, base one, and targets overlap. You can exclude regions that you don't want to include for the PCRs, and pair okay region list, so you can add any. Here is the instructions for that one, and you can select anyone or now we don't need to that one. And here the general primer picking conditions, that are the white bars here mentioned that the primer size, here they are mentioned that from 80 to 20 minimum, and this is the optimum 20 base pairs, and the maximum 23. You can also change here 30 base pairs or 18 to 22 24. This is the melting temperature for the primer. This is the mostly 57 to 59, minimum 57, optimum 59, and maximum we can select that. You can select any from 50 to 60, and the product and the primer GC concentrations, that is the 30 to 50 optimum, and 70. That shows the main regions, means that you have the wider acceptability of the primers from the 30 to 70, so that these are the guidelines. So for example, when you have, it will, if you have to select the pick primers, by picking the primers, it will automatically select the all the primers that are according to these conditions. Here we can see the left primer, forward primer, reverse primer, and the start of the position of the nucleotide, that is the 46, from the start of the 46 base pairs, and the total length of the left primer is the 20 base pairs, so that it is good one. And the TM is around 59, that is also less than 60, and the GC content is also within the optimum conditions around 40 to 60, that is the 55%. And there is no hairpin secondary structure are available in this one. And the right primer, that is also start from the 291, and the total length of the primer is 18 base pairs, and the melting temperature is also almost similar one, 59.8 for right primer, and 59 from the left primer, and GC is also somehow okay. And then we will, total sequence size is the 2420, and the included regions is okay. Here you can see that there is the sequence, this is the total sequence of the, that we have the pasted, and the left primer, that is starting from this one. That means it will start from this one to amplify this one. That is the region that will be amplified. And here is the reverse primer that will start from this one. And we have to see that, know that which region is we have to amplify. If your region is in this one, that is okay. And if you, there is also the additional oligonucleotides that are primer, they have automatically selected the top one, and they have given to us with the primer the best one. The next we can also check that the next one and the is this one. And then you can see that it is around starting from the 43 to 57. If your region is around only in between, maximum from the 241 to 3, in between 394, you can also select this one, because it will the shorter one, and it will not good, it will be good if you are the specific regions and you don't need to amplify the whole region, so that you can select the any this one. And I hope, and you can, this is the statistics, and you can select according to the melting temperature 2156, so that you can select the any primers. And so that this is the, this is the sequence of the forward primer, and this is the sequence of the reverse primer. You can copy, only copy this one, and you can send to the company for the primer designing. Here we will see that this is the basic primer 3 output, and this is the design of the primers. And if we see the the second one, the primer 3 plus, this is also showed the minimum parameters. You can select that this is the advanced one, and here you can also paste the sequence. Here this is also the detection. The detection is generally used for the PCRs and the cloning. If you are have to clone the regions, that that means from the start of the sequence base pairs at the end of the sequence, then you can select the cloning option for the primer. And then for the sequencing, for sequencing the regions, you can check the primer and the primer list primer check. Here for the generally for the primers PCR, here you will see the only detection. This is the general settings. This is also for the primer size, primer temperature, GC ratio. You can also manipulate this one, and these are the advanced setting. You can see that here you can, if you have the, if you have any excluded region you want to add here, any targets included region, here you can only to the pick primers, this one. And here you can see that this primer and the total length of this one, melting temperature, GC ratio, so that if one, and it is also showing that this region, the start of the forward primer, this is the start of the reverse primer from this one, and this is the region you want to amplify. Here is the primer pair 2, 3, 4, and according to these conditions and the specific regions you want to amplify, you can select these primers, so that these are is the over one. I hope you will know about the some basics techniques or some basic guidelines to know how, somehow basics for the primer designing. Thank you so much.